Welcome to a run-through of the AS Level Biology OCR Breadth in Biology paper from May 2016. I hope you find it useful. Major goal is that you should understand how to approach the questions a bit better and maybe understand how to answer them a bit better. Good luck with your exams. So the paper begins with 20 multiple choice questions. Microscopes vary in their magnification and resolution. So magnification is the number of times bigger that the image is than the object, and resolution is about clarity. It's the ability to perceive two things as being two things rather than one thing. So resolution is the minimum distance apart that two objects need to be for you to perceive them as two objects rather than one. Now for a light microscope, that's around half the wavelength of what you use to illuminate your specimen. So light is around 400 nanometers at its shortest visible wavelength, so half of that is around 200 nanometers. So the resolution for a light microscope is 200 nanometers. The resolution for an electron microscope is around 0.2 of a nanometer. So you've got 1,000 times greater resolution with an electron microscope than you do with a light microscope, which is why you can see so much more detail. So looking at this question, things you need to eliminate. So first of all, the line A. Line A gives you a magnification of only 10,000 times for the transmission electron microscope and scanning electron microscope 50,000, so we can cross off A. Now B, it tells you that the maximum magnification for a light microscope is only 400 times. That's definitely not true, so we can cross that off. Which leaves us with just C and D. Now C tells us that the resolution for a light microscope is 200, which is correct. The resolution for a transmission electron microscope is 0.2, which is correct, and 0.2, which is correct. And they tell you for D that the resolution is 100 nanometers, and that's wrong. So the correct answer here is C. So carbohydrates such as starch are made from monosaccharides joined together. So mono means one. Remember in biology we've got mono, one, di, two, tri, three, and poly, many. And the suffix saccharide means it's a sugar. So which of the bonds joins monosaccharides together? Well, this would be a glycosidic bond. And the reason for that is that an ester bond joins together a glycerol to fatty acids. A peptide bond joins amino acids together to make a peptide and then a protein. And a phosphodiester bond joins together nucleic acids in a chain to make DNA or RNA. Whereas a glycosidic bond is made by a condensation reaction between two sugar units. So here we might have alpha glucose with the OH on the one carbon, and it would form a glycosidic bond with another glucose joining the 1 and the 4 carbon together, eliminating water because it's a condensation reaction, and then putting the oxygen in the middle of the glycosidic bond. So the answer here is B, a glycosidic bond. So some inorganic ions have roles in enzyme control reactions. Which of the rows A to D is correct? So the first thing is you can exclude the ions which are incorrect. So zinc, remember, forms 2+ ion, not a 1+, so you can exclude that, and you can exclude the chloride ions because that forms a 1- ion. So this leaves us with the options of D or A. And unfortunately you just need to know this, and so you need to know that the chloride ion is a cofactor for amylase, and that zinc is a prosthetic group for carbonic anhydrase, which causes the making of hydrogen carbonate and the Bohr shift. So the answer here is D. So now it talks about what of the following statements about the nature of the genetic code is incorrect. So it is a degenerate code, it is a triplet code, a sequence of 3 nucleotides codes for 1 amino acid. It is universal, well, as long as you ignore mitochondria and some ciliated protozoa, which have a couple of triplets which are different, but it is not overlapping. A sequence of 3 nucleotides codes for 1 amino acid, and then the next sequence of 3 nucleotides codes for the next amino acid. So it's not overlapping, so the answer is C. C is incorrect. And then ventilation involves various parts of the mammalian respiratory system, which are the following statements describes inhalation. Now, in inhalation, you have to increase the volume of the thorax, because if you increase the volume, you decrease the pressure, and when you decrease the pressure, air will move into the lungs. So by increasing the volume, we would move the rib cage up and outwards, which means that it has to be either A or C. The external intercostal muscles would contract and the diaphragm would contract, because remember, the diaphragm is a domed shape when it's relaxed, and when it contracts it forms a flattened sheet, which would increase the volume of the thorax so that air would move in, because there is a larger volume, therefore a lower pressure. So the answer here would be C. So which of the following structures A to D are found in both prokaryotes and in eukaryotes? Well, a cell wall made of peptidoglycan is only found in prokaryotes. A circular genomic DNA, so a single circular chromosome, is found in prokaryotes. A nucleus surrounded by a nuclear membrane is only in eukaryotes, which leaves us with D, ribosomes, which are the site of protein synthesis in both prokaryotes and eukaryotes, so the answer is D. Now on to the pressure changes in a mammalian cardiac cycle. Now remember, when you're doing this sort of thing, or looking at this sort of graph, remember you've got atrium, then a valve, then ventricle, then a valve, and then the aorta. Remember that valves are pushed open by the flow of blood, and they're pushed closed by the flow of blood, not that they open and allow the flow of blood through them. Remember that blood will flow from a higher pressure to a lower pressure. So if you look at the dotted line for ventricular pressure, over this region here, the pressure in the ventricle is lower than the pressure in the atria, which means that blood will be flowing through the open valve from the atrium into the ventricle. If you look at this section here, which is between 0.2 and 0.3 of a second, you get a big increase in ventricular pressure. So at this point here, you would get the closing of the atrioventricular valve, because the blood is being pushed back towards the from the ventricle towards the atrium. That pressure will continue to increase until the pressure becomes greater than in the aorta. And at that point, the valve will be pushed open between the ventricle and the aorta, and blood will flow from the ventricle into the aorta, so the atrioventricular valve will open at this point here. And the question says, which of the following time periods A to D shows ventricular systole, and ventricular systole will be occurring between 0.3 and 0.2 of a second, so the answer here is B. So measles is a serious disease that can be prevented by vaccination. The chart below shows the measles-containing virus, MCV, coverage, and the annual reported cases of measles between 1980 and 2013. So on this side we've got the number of cases, on this side we've got the coverage by the vaccination. And then they give you a series of statements which is a correct interpretation of the chart. So firstly, they say that an increase in human herd immunity resulted in fewer cases from measles. Now, this may be true, but you can't say that the increase in human herd immunity resulted in fewer cases. You can see that there is a herd immunity when you get to 90% or higher than 85% vaccination, but you can't see that an increase in human herd immunity resulted in fewer deaths, so you can't say that it's A. The highest number of measles cases occurred when the coverage was at its lowest, while the highest number of cases is in 1981, and that is not when there's the lowest coverage, because the lowest coverage is in 1980, so that's incorrect. D, there's a positive correlation between the number of measles cases and the MCV coverage. Well, there isn't. There's a negative correlation, because as you increase the coverage, the number of measles cases falls. So there's not a positive correlation. Which leaves us with C. A 90% MCV coverage resulted in fewer than 500,000 cases of measles each year. So if we look at 500,000 cases, which is here, so it's this line, and we can see that when we have 90% up here, we can see that it's fewer than 500,000 cases, so the correct answer would be C. Two different fields, field G and field H, were sampled for 3 common species of wildflower. The results are shown below. So the species were daisy, dandelion and buttercup, and you've got 2 fields. And then from looking at the data you can see that you've got an awful lot of buttercups in field H, but very few dandelions and daisies. Whereas in field G you've got approximately the same number of daisies, dandelions and buttercups. So the species richness of all of them is the same. There are 3 species in both, so neither has a greater species richness. Now species H has a greater species evenness. Well, that's not true, because most of the individuals that are present are one species, which is the buttercups, whereas species G has a far more even distribution, in that about 1/3 are daisy, 1/3 are dandelions and 1/3 are buttercups. So B is not true. Now field G will have a greater Simpson's diversity index, because they've got the same number of species in both, but there's greater species evenness in field G than there is in field H, so that there's a greater diversity in field G, so the answer here is A. So which of the following options, A to D, lists the 3 domains of life? Now this is the idea that life can be split into 3 domains, 2 of which are prokaryotic and 1 of which is eukaryotic. So this means we can exclude C and we can exclude D, because D gives you all eukaryotic and C gives you 2 prokaryotic, which means leaves us with A and B. And the answer is A, because the archaeans are a domain of life, as are the bacteria, as are the eukaryota. So the answer is A. Now, a student investigates some solutions X, Y and Z using paper chromatography, and the result is shown below. Now, which of the following options is the RF value of Z? So RF means relative front. So it's the distance moved by the spot relative to the distance moved by the solvent front. So you need to get out your ruler at this point and measure the distance travelled by the spot and divide by the distance travelled by the solvent front. And this means you can compare chromatograms which have been run for different lengths of time. So for Z, you need to measure the distance travelled by the spot and then divide by the distance travelled by the solvent, and that gives you the RF value. And if you do that, you realise the answer is A, 0.63. So pressure varies in different parts of the mammalian circulatory system. So you've got pressure in the blood in the aorta, and remember the aorta is exiting from the left ventricle, so blood will be at a very high pressure in all of them. And this is the same in A to D. Now, the vena cava is where the blood goes back into the right atrium of the heart, having circulated the body. Now, it's coming from the capillaries, where there's a very low pressure, and the blood is at a very low pressure in the vena cava, and the reason it flows at a reasonably high velocity is because you've got a very large diameter lumen, so that you've got comparatively less resistance because you've got such a big lumen in the vena cava. So the blood is at a low pressure in the vena cava. Now, the tissue fluid has been forced out of the capillary by the higher hydrostatic pressure than the oncotic pressure between the tissue and the blood plasma. So the tissue fluid is at a low pressure. Excess tissue fluid flows into the lymphatic drainage system, and that additionally is at a very low pressure. So the answer here is C. So we've got a diagram of a potometer. A potometer measures the rate of water uptake into the stem of the plant, and it's used as an indicator of the rate of transpiration, which is the evaporation of water from the spongy mesophyll and the palisade mesophyll from the leaves, and that water is then replaced by water being drawn up the xylem in the cohesion tension theory. Now, which of the following options is a precaution that's not needed when setting up the potometer? Excess water must be removed from the leaves before readings are taken. Well, yes, you definitely want to do that because you wouldn't want the spongy mesophyll on the stomata to be blocked by water droplets on the surface of the leaf. The root should be cut underwater. Well, the reason you do that is to prevent air bubbles from entering the xylem. Therefore, if it did, then it would break the continuous flow of water in cohesion tension. There should be no extra air bubbles. That's correct. You don't want any extra air bubbles in here. And the screw clip must be opened while taking the readings. Well, if you opened the screw clip, water would come down and water would push the air bubble out. So B is definitely an option that you shouldn't take. So they show you a stage in mitosis and then ask you which of the following options is the stage of mitosis shown above. Now remember, mitosis consists of prophase, metaphase, anaphase and telophase. Prophase, they appear. P is for appear. Metaphase, they line up on the middle. Anaphase, they get pulled apart. and in telophase you form 2 new nuclear membranes. So in prophase the condensed and duplicated chromosomes appear as 2 chromatids joined together by a centromere forming a chromosome. In metaphase they line up on the middle. In anaphase the centromeres split and the 2 chromatids are pulled to the opposite poles, and in telophase you form 2 new nuclear membranes. And what's shown here is metaphase B. So now we've got a simplified structure of an antibody. When you're looking at an antibody, you should identify A as the variable region, which is complementary to the antigen. And then you should identify B as bonds between the polypeptide chains. And C you should identify as the hinge region. And the hinge region enables the distance between the antibody binding sites to vary. So this hinge region C means that the distance between the antigens and the antibody binding sites therefore can vary. So the answer here is C. So a group of students were investigating the diffusion of molecules across membranes using a model cell. Now into the model cell, which is an artificial cell membrane, they put a solution containing starch and amylase. They then used biochemical tests to identify the molecules that were present at the beginning and the end of the experiment, in both inside the model cell and outside in the extracellular fluid. So if they've put starch, remember is a polysaccharide of alpha glucose joined together by glycosidic bonds forming amylose and amylopectin. Now starch is then going to be hydrolyzed, the glycosidic bonds are going to be hydrolyzed by amylase, which is an enzyme, so it's a protein. And it's going to be hydrolyzed into maltose, which is a disaccharide, it's a reducing sugar. Now the starch is going to test positive with iodine, it's going to form a blue-black colour. The amylase is going to test positive because it's a protein, therefore it will test positive with the biuret reagent. And the maltose is going to test positive because it's a reducing sugar with the Benedict's reagent. So they then show you a table of the molecules that are present. So having a look at the first row, well, the Benedict's tests test positive in the extracellular, they're comparing the correct results for the cytoplasm at the beginning and the extracellular fluid at the end. Now, at the beginning, there's going to be no maltose in the cytoplasm, so A is correct, and B also has got no maltose in the cytoplasm, whereas they do have maltose in the extracellular fluid, and the reason why it'd be there is because it will have diffused out because the digestion would have occurred inside the model cell. So the starch would have been hydrolyzed to maltose, and then the maltose would have diffused out. So there's no maltose present at the beginning, but there is maltose present in the extracellular fluid at the end of the experiment. So this means we can discard C and D because they're definitely wrong. The biuret test is going to be testing for the presence of the protein. So there's definitely protein in the cytoplasm at the beginning because there's the amylase that you've put into the model cell. But the amylase will be too large to diffuse across the extracellular fluid, so it's definitely not B, so we can rule out B. Now the final one, which is unnecessary because the answer is already definitely A because we've excluded all of the others, The final one is the iodine test. Well, the iodine test will test positive in the cytoplasm for all of them, because starch will be in the cytoplasm because you put it there, whereas the starch is too large to get across the membrane, so it won't be in the extracellular fluid. But the answer we've already worked out here is A. So which of the cells below, represented by the cubes A to D, has a surface area to volume ratio of 3:1? So remember, a cube has 6 sides. Remember, a dice has 6 numbers on it, so a cube has 6 sides. So to work out the volume of cube A, it's 1 times 1 times 1, which gives you 1, and the surface area is 1 times 1 for the surface area of one side times 6. So this gives you a surface area to volume ratio of 6:1. Cube B has got a volume of 2 times 2 times 2, which gives you 8. And it's got a surface area of 2 times 2 times 6, which gives you 24. So it's got a surface area to volume ratio of 24:8, which is 3:1. So the answer here is B. The others, it's for the cube sized 4, it's 96:64, which is 3:2. And then for cube D, it'll be 6 times 6 times 6, which gives us 216. And then the volume and the surface areas are going to be the same. So this gives us 1:1. So the answer here is B. So a diagram of a stained blood smear is observed under a light microscope. Which of the structures A to D is a neutrophil? Well, the answer, you just have to know this, I'm afraid, is B, because the neutrophils have distinctive lobed nuclei. So when you see the nucleus, which is black in the middle, when you see this distinctive lobed shape, you know that you're looking at a neutrophil. So the answer is B. And which of the following options, A to D, is a primary defense mechanism against pathogens? Now, neutralization and agglutination are caused by antibodies, and phagocytosis is part of the non-specific immune response. But blood clotting is a primary defense mechanism because it prevents pathogens from getting in, or prevents more pathogens from getting in, by forming a scab because the blood is clotting. Therefore, it's a primary defense mechanism because it stops more pathogens from getting in. So the answer is D. So pathogens cause disease and are transmitted from individuals to individuals in a variety of ways. Which of the rows in the table is correct? So first of all, look at the type of pathogen. It tells you that TB, D, is caused by a protoctist, which is not true, it's caused by a bacterium. They tell you that malaria is caused by a bacterium, which is not true, it's caused by the Plasmodium parasite, which is a protoctist. So it can't be C and it can't be D. Then they tell you that HIV/AIDS, which is a virus, that's correct, is caused by indirect contact, and that's not true, it's caused by the transmission of the virus in blood or bodily fluids directly between individuals. So the answer here is A, because athlete's foot is a fungus and it's transmitted by direct and indirect contact. So the answer is A. So now on to the longer response questions. So they show you some apparatus used in an experiment investigating water potential in potato tuber tissue. So the discs were placed in boiling tubes containing sucrose solution of different concentrations for 4 hours. Then they measured the percentage change in mass of each disc. So the independent variable here is the concentration of the sucrose solution into which the potato discs were placed, and the dependent variable is the change in mass of the potato discs. When you put the potato disc, so we draw a potato cell here, when you put a potato cell into a solution of less negative water potential, then the water will enter by osmosis. As a result of entering by osmosis, the mass of the potato tissue will increase. So when we have the highest water potential, which is a water potential of 0 because we've got no sucrose because it's distilled water, then we have the highest increase in mass. As we decrease the water potential because we have more sucrose dissolved in the water, then the percentage increase of the potato discs reduces because there's less of a water potential gradient between the external solution and the cytoplasm within the potato. At 0.2 molar sucrose, the potato only gains 2.5%, and at 0.3 molar sucrose it loses 3%. So somewhere between 0.2 and 0.3 is the point where the water potential of the external sucrose solution is the same as the water potential of the potato, because at that point it hasn't gained or lost mass, so that we would say that the sucrose solution at that point would be isotonic with the water potential of the potato cell. Now when we put the potato cells into strong sucrose solution, then at that point the water potential is more negative in the sucrose solution than it is in the cytoplasm of the potato, so the water leaves by osmosis, going down its water potential gradient into the more negative water potential in the solution. Therefore the potato loses mass, and the stronger the sucrose solution, the more mass the potato loses, because the bigger the difference in water potentials. Now state 2 details of the procedure that must be followed to obtain valid results. Now valid results means that the experiment is actually testing the phenomenon that you say it's testing. So to ensure validity, you should do things like make sure that the potato discs are all the same size, because that would affect the rate of diffusion, therefore the rate of osmosis. You need to compare like with like, so you should use the same variety of potato. There should be no skin on the potato. To make your results more accurate, you should remove and therefore more valid in this case, therefore you should blot off the excess water before weighing. You should use the same number of discs in each solution, the same volume of sucrose solution, never write amount ever in anything. You mean either mass or volume or concentration. The same temperature, and to cover the tubes to prevent the evaporation of water. So explain how a student could use the data in the table to determine the water potential of the potato tuber tissue. Now the concept thereafter here is that you realise that the water potential of the potato tuber tissue determines how much water it's gained or lost in each of the different concentrations of sucrose. So for instance, you could plot a graph with the independent variable, which is the concentration of sucrose, and then you could plot the percentage change in mass, and then when it crossed the axis, you would know that at that point the potato had neither gained nor lost mass, so the water potential of the external solution would be the same as the water potential of the potato tissue. So at that point you'd have to look up the sucrose concentration in a table and look up what the water potential of that sucrose concentration was, to then tell you what was the water potential of the inside the cell. Another way that you could have determined this more accurately is, remember that you've got between 0.2 and 0.3 is where you get the change from gaining mass to losing mass. So if you did more measurements between 0.2 and 0.3, so say you did 0.21, 0.22, etc., then you would be able to more accurately determine the water potential of the potato tuber tissue. But in your answer, do remember that they're mainly looking for you to demonstrate that you understand the concept of why the potato is gaining or losing mass, and that's because of its water potential relative to the concentration of the external solution. So the answers that they were looking for here was, firstly, the idea that no change in mass occurs when the water potential of the sucrose solution equals the water potential of the potato tissue, that there's no change in mass between 0.2 and 0.3 moles per decimeter cube of sucrose, that you could plot a graph of concentration against percentage change in mass and then look at the point at which there was no change in mass. Alternately, you could carry out the experiment again with more concentration intervals between 0.2 and 0.3 and then look up the water potential of the sucrose solution in a table, and then that would give you the water potential of that solution. Okay, then they show you a diagram of a cell from a potato tuber disc that was placed in 0.45 molar sucrose solution. So we've got 0.45 molar outside, and the potato tuber disc has lost mass. The reason it's lost mass is that there's a more negative water potential outside, so that the water has moved by osmosis, and the volume of the cytoplasm has reduced. Now the cell membrane remains around the cytoplasm, but the cell membrane has come away from the cell wall, which is a situation known as plasmolysis. So Y is the membrane, definitely. X is the cell wall. V would be the cytoplasm. Z would be the tonoplast, which is around the outside of the vacuole. And U would be the vacuole. So it then says identify the parts of the cell. So X is the cellulose cell wall, Y is the plasma membrane, and Z is the vacuole membrane or tonoplast. Now what will be found at W in the immersed cell? So this is making sure that you understand the concept that cellulose is permeable to sucrose, to the sucrose solution, sorry. So what will be found at W, which is the gap between the cell membrane and the cell wall, what will be found there is the sucrose solution. In fact, it will be the 0.45 molar sucrose solution that you've put the cell into, because that sucrose solution can cross the cellulose cell wall because it's permeable. So what will be found at W will be the sucrose solution. So halophytes are plants that have the ability to live in soils with a very low water potential. In the UK, these plants form part of salt marsh communities. Suggest and explain, and suggest means you can come up with anything that is biologically possible, and explain, say why this factor causes it, or causes the absorption of water. So suggest and explain how the root hairs of halophytes are able to absorb water by osmosis from the soil. And they've told you that the soil has a very low water potential. So how could the halophyte root hair absorb water from a very negative soil water? Well, the way they can do that is by making the cytoplasm of their cells a more negative water potential. So they must make it a more negative water potential in order for them to absorb water by osmosis across the root hair. And then your suggest bit is to say how that could occur, and that could occur by the active transport of ions and salts across the membrane of the root hair cell and into the cytoplasm or the vacuole of the root hair, therefore lowering the water potential, therefore making it possible to absorb water from the soil water, which has a very low water potential because of the saltiness of the salt marsh. So what they were looking for, the key concept, is that there had to be a lower water potential inside the root hair cell than in the soil water. How you could achieve that would be through active transport of mineral ions or salts into the root hair cells, or alternately that the cells could be a store of mineral ions or a store of solutes, which would also lower the water potential. But the major message was that you had to say that you understood how you understood that the root hair cell had to have a more negative water potential than the soil in order to be able to absorb water by osmosis from the soil. So even the smallest DNA molecules are very long. A kilobase is a unit equivalent to 1,000 base pairs of DNA. 1 kilobase has a length of 0.34 micrometers. The DNA in the nucleus of a cell from a fruit fly is 5.6 centimeters long. Calculate the number of kilobases in the DNA of the fruit fly, show your working, and give your answer to the nearest whole number. So they've told you it's 5.6 centimeters. First thing to do is always to convert it into SI units, which is 56 millimeters. Remember that there are 1,000 micrometers in a millimeter, so this gives us 56,000 micrometers. Now they've told you that 1 kilobase has a length of 0.34 micrometers. So to work out how many kilobases there are, you divide 56,000 by 0.34 micrometers, which gives you the answer of 164,705.88. But they want the answer to the nearest whole number, so the answer is 164,706. Now, the DNA of the fruit fly was analysed and 22% of the bases were adenine. What percentage of the bases were guanine? Well, if 22% of the bases are adenine, remember adenine forms 2 hydrogen bonds with a thymine, which would mean that 22% would be thymine. Now adding the two together, that gives you 44%. Remember the other base pair is a guanine, which forms 3 hydrogen bonds to a cytosine. So 100 minus 44 would give us 56% of the bases would be guanine and cytosine. Of those, half of them are going to be guanine, half of them are going to be cytosine, so 56 divided by 2 gives us 28%. So the answer was 28%. So a DNA molecule contains polynucleotide strands. Individual nucleotides are joined together. What type of reaction takes place when 2 nucleotides in a single polynucleotide strands are joined together? So this is referring to the bond that forms between the sugar of one and the phosphate of another. So this is forming a phosphodiester bond, and the type of chemical reaction that takes place is a condensation reaction because it produces water. Name the chemical released when the bond is formed between the 2 nucleotides. Well, again, it's water. And then A DNA molecule contains two polynucleotide chains. Describe how these chains are held together. Remember that you've got a phosphate, a deoxyribose sugar, and then a nitrogenous base, of which there are purines and pyrimidines. Adenine and guanine are purines. The purines have 2 rings. And cytosine and thymine are pyrimidines, and the pyrimidines have 1 ring. Now, an adenine and a thymine form 2 hydrogen bonds, and a guanine and a cytosine form 3 hydrogen bonds between the nitrogenous bases. So the answers they were looking for you to put here were that there are phosphodiester bonds in the backbone between the phosphate and the deoxyribose sugar, that there is hydrogen bonds between the bases or between the chains, that are purine bonds to a pyrimidine, an adenine to a thymine, a guanine to a cytosine, and that 2 hydrogen bonds form between an adenine and a thymine, and 3 hydrogen bonds form between a guanine and a cytosine. So the Sumatran rhinoceros, which is Dicerorhinus sumatrensis, is a rare member of the family Rhinocerotidae. Right, that's enough of my appalling pronunciations. So they give you a binomial name. They give you the scientific name. And when you see this, you need to think, well, this is the genus, because it's got a capital letter, it's in italics, and then this, following it, is the species name. Just like Homo sapiens, Homo, remember, is the genus, sapiens are the species. These rhinos are now critically endangered, with only 6 substantial populations in the wild, 4 in Sumatra, 1 in Borneo, and 1 on the Malay Peninsula. They live in forests. Their number is difficult to determine, but they are estimated to number less than 100. So complete the table below showing the classification of the Sumatran rhino. Now, they don't expect you to know how to classify the Sumatran rhino, and they're not asking you to do that. What they're asking you to do is to remember that there's a hierarchy of classification, which goes kingdom, phylum, class, order, family, genus, species. Now, if you want to remember a mnemonic, it's King Philip came over for good sausages. You should, at the very least, remember that rhinos, and hopefully you're familiar with a rhino is an animal, so it's in the kingdom of Animalia. Now, the level below phylum, well, that's class, King Philip came, order, over, for, good, genus, and then at the bottom, species, you should know from Homo sapiens, so you should know that this is the species, which is the second word, which is sumatrensis. Now, the name Dicerorhinus sumatrensis, last one I hope of these, is part of the binomial naming system. So suggest an advantage of using this system rather than saying its common name. So it's saying, why do scientists use a binomial naming system rather than just calling them whatever they want? And the idea here is that this is a name that is universally recognized. It's the same in all languages. You therefore know which species and genus it belongs to, because many different civilizations can have a different common name for the same organism. So by having a binomial name that is the same in all languages, then you know that you're always talking about the same organism. So suggest 2 reasons why this species is critically endangered in the wild. So you could come up with any suggestion that would have caused these species to come to very low levels. So suggestions that would have been acceptable here would be things like hunting, because the horn of the rhino is regarded as being very desirable by some cultures, so they're hunted or poached. The loss of rainforest, because the habitat has been cut down through deforestation. The loss of food in the rainforest habitat, or alternately climate change. So all of these are suggestions as to why this species could be critically endangered in the wild. Then it goes on, the remaining populations are all small and are scattered in isolated areas. These are factors that might speed up the extinction of the species. So in your answer, you've got to say why having small populations and scattered, isolated populations, which might speed up the extinction of the species. So the points they're looking for here with the small population is to think that small populations lack genetic diversity. And because they're scattered in isolated areas, those small populations are unable to interbreed. So that as a result, if you got a disease or a change, then there's very little genetic diversity, so they're very susceptible to changes in the environment. And they're also very susceptible to disease. So the answers they were looking for here was, it may be hard to find a mate because you've got very isolated populations, and that inbreeding leads to a low genetic diversity because there's a small gene pool, and gene pool, remember, is the total number of alleles in a population. So this might lead to a genetic bottleneck. As a result of having a small gene pool, it's less likely to cope with environmental change. They could all be wiped out by the same disease. Because they're small populations, they're more vulnerable, and they're isolated populations, they're more vulnerable to poachers or predators. And a natural disaster, such as a volcano or a flood, could wipe out one of those small populations. Finally, captive breeding programs have been unsuccessful. Suggest one other way in which zoos can contribute to the conservation of the Sumatran rhino. While the other ways it could do is through education and raising environmental awareness of the problems of the destruction of habitat or the problems of climate change. And additionally, they can also promote support for in situ conservation projects and carry out research. So in mammalian blood, oxygen is mainly transported combined with hemoglobin as oxyhemoglobin. The presence of hemoglobin greatly increases the oxygen carrying capacity of the blood. So 100 centimeter cubed of plasma contains 0.3 centimeter cubed of oxygen when fully saturated, whereas 100 centimeter cubed of blood contains 20.1 centimeter cubed of oxygen when fully saturated. Calculate the percentage increase in oxygen carried in fully saturated blood compared with the oxygen carried in fully saturated plasma. Well, the percentage increase would be the saturation of oxygen in blood minus the saturation of oxygen in the plasma divided by the oxygen in the plasma, which gives you 19.8 divided by 0.3, which gives you 66, multiplied by 100 to give you a percentage, gives you 6,600% more oxygen in fully saturated blood than in fully saturated plasma. So the answer here is 6,600 times, because you've got to, in order to work out the percentage increase, you've got to work out the difference between the blood and the plasma and divide by how much oxygen is in the plasma. Now, hemoglobin is found in erythrocytes. Unlike other vertebrates, the mature erythrocytes of mammals lack nuclei and other membrane-bound organelles. So it lacks things like mitochondria, which are the other membrane-bound organelles that they're referring to here. So explain one advantage and one disadvantage of the lack of nuclei and other membrane-bound organelles. So the advantage is that through having no nucleus, they can carry more hemoglobin and also have a better surface area to volume ratio, and additionally they are able to squeeze through capillaries. So advantages are more space for or contain more hemoglobin or oxygen, therefore they can also squeeze through the capillaries more easily. The disadvantage is that with no nucleus they can't make their own proteins, and with no mitochondria they can't carry out any respiration. So they'll have a limited lifespan because they can't carry out protein synthesis, because they don't have any DNA to give you the codes to make the proteins. They can't reproduce, they can't undergo mitosis because they don't have any DNA. They have no respiration by the mitochondria. They've got no aerobic respiration, they can only carry out anaerobic respiration. Okay, then on to viruses do not use erythrocytes as host cells, whereas the Malaria pathogen Plasmodium falciparum spends part of its life cycle inside erythrocytes. Suggest why. Now, they've already told you that erythrocytes don't have nuclei, therefore they can't carry out protein synthesis, so they wouldn't have any ribosomes to carry out protein synthesis. So if the virus wanted to replicate itself, then being in a erythrocyte is a very bad idea. Whereas the Plasmodium parasite has got its own machinery because it's a living organism, it's a living cell, it's a protoctist. So the Plasmodium parasite is able to reproduce able to use the red blood cell as a host, and then use the resources inside the red blood cell, and also it hides from the immune system of the host. So answers that they were looking for here is that the virus is unable to replicate on its own outside of the host cell because it requires the host cell's DNA and ribosomes for protein synthesis. Now, the erythrocyte is not going to have the ribosomes for protein synthesis, so the virus shouldn't replicate within the red blood cells. Whereas the Plasmodium parasite is using the red blood cell to hide from the immune system. It's completing its life cycle by using the contents of the red blood cell as a source of food for the growth of the Plasmodium. So in this answer you needed to link the absence of all of the nuclei and the membrane-bound organelles to why the virus wouldn't reproduce inside an erythrocyte, whereas the Plasmodium would be able to hide from the immune system and also use the red blood cell as a source of food. So explain why erythrocytes do not make use of the oxygen that they are transporting. Well, remember the erythrocytes don't have mitochondria, they can't carry out aerobic respiration, so they won't be consuming the oxygen. Additionally, the erythrocytes are just in the blood that is flowing, so there's just mass flow of the erythrocytes, that the movement of the erythrocytes does not require any energy to be expended by the red blood cells. Additionally, the oxygen is bound to oxyhemoglobin. So all of these points were relevant. that the oxygen is bound to the hemoglobin. They don't have any mitochondria, therefore they don't carry out any aerobic respiration, and they're moved by mass flow, so it doesn't need ATP to move it. Now, oxygenated blood returns from the lungs to the heart before being pumped around the body. Normal cardiac output's about 5 decimeters cubed per minute, and 100 centimeter cubed of blood contains 20.1 centimeter cubed of oxygen when fully saturated. So calculate the volume of oxygen being transported to the tissues per minute. So this requires you to know that 100 centimeters cubed is 0.1 of a decimeter cubed. Therefore, there would be 50 100 centimeters cubed in 5 decimeter cubed. So there would be 50 times 20.1 centimeters cubed of oxygen, which would give us the answer of 1005 centimeters cubed of oxygen. So the answer here is 1005, because there are 100 centimeters cubed is 0.1 of a decimeter cubed, therefore there are 50 100 centimeters cubed in 5 decimeters cubed, therefore there is 50 times 20.1, which gives us 1005. With reference to the structure of blood vessels, explain why oxygen is not released until the blood reaches the capillaries. So, with reference to the structure of blood vessels, wants you to think about the fact that a capillary is only 1 cell thick, and that's a very thin epithelial cell. And that gives you a very short distance for the diffusion of oxygen off the hemoglobin into the tissues, where there's a lower partial pressure of oxygen because the oxygen is being turned into water. But with an artery and an arteriole, you've got a tunica intima, then you've got a tunica media, and then you've got a tunica externa. So you've got a much thicker wall with a much greater distance for diffusion. So the answer wanted you to refer to the structure of the blood vessels, saying that the arteries and arterioles have a thick wall in comparison to the much thinner wall, which is only 1 cell thick, of the capillary. And that, therefore, there would be no diffusion through the artery wall, because the diffusion distance would be too large, and that this diffusion would occur through the capillary wall because there's a short distance for diffusion, because the epithelial cell is very thin. So figure 24 shows the oxygen dissociation curves at different carbon dioxide concentrations. So at a low partial pressure of oxygen, you've got this curve, and a high partial pressure, this curve. Now, what name is given to the change in the oxygen dissociation curve due to increasing carbon dioxide concentration? Well, you should know that this is called the Bohr shift, and that's caused by the carbon dioxide. Now, what causes that is that the carbon dioxide, remember, is produced in the mitochondria, and it's produced by the decarboxylation of carbohydrates and fats. The carbon dioxide diffuses out of the mitochondria into the plasma and then diffuses into the red blood cell. Now, once it's diffused into the red blood cell, it joins with water, and this is catalyzed by carbonic anhydrase to produce hydrogen carbonate. Now that then dissociates to produce the hydrogen carbonate ions, which diffuse out because there's lots of them and there's fewer in the plasma, and that's balanced by the influx of the chloride ions, which is known as the chloride shift. Now, the dissociation of the hydrogen carbonate produces hydrogen ions, which lowers the pH. Those hydrogen ions then join with oxyhemoglobin and produce hemagglutinic acid. And that causes the dissociation of the oxygen. So the oxygen comes off the hemoglobin and diffuses into the cells where it's used in the mitochondria for aerobic respiration. Now, the more you respire, the more carbon dioxide will be produced, which means you'll produce more hydrogen ions, which will join with more of the oxyhemoglobin, which will cause more of the oxygen to come off and therefore reduce the affinity of hemoglobin for oxygen. So going back to the question, what name is given to the change in concentration in the dissociation curve? Remember, that was the Bohr effect or Bohr shift. And then letter T indicates the partial pressure of oxygen in actively respiring tissues. These actively respiring tissues are reducing lots of carbon dioxide. And then explain why the blood offloads more oxygen to actively respiring tissues than to resting tissues. Well, that's because there's more carbon dioxide produced by the actively respiring tissues than by the resting tissues. So if we look at the point T, we can see that for the same partial pressure of oxygen, the actively respiring tissue, which has got more carbon dioxide, is going to have more oxygen dissociate from the hemoglobin. So this extra oxygen will come off because of the higher partial pressure of carbon dioxide produced by the actively respiring cells. So your explanation needs to talk about actively respiring tissues are going to have a higher partial pressure of carbon dioxide, which will lower the affinity of hemoglobin for oxygen, because the extra carbon dioxide causes the dissociation of the carbonic acid, increasing the number of hydrogen ions, lowering the pH, which leads to the release of oxygen because it decreases the affinity of hemoglobin for oxygen, and therefore more oxygen will be released at the same partial pressure of oxygen, so that you will get a bigger dissociation when you have a higher carbon dioxide partial pressure in the tissues than when you have a lower carbon dioxide partial pressure. So the following statements summarise the results from experiments designed to discover more about the translocation of organic materials in the phloem. So any increase in sugar content of the leaves is followed by a similar change in the sieve tube contents. The rate of transport increases with increasing temperature, reaching a maximum of 25 degrees before decreasing at higher temperatures. Now that would tell you that there was something to do with enzymes and something to do with the activity of those enzymes. Translocation stops when stems are treated with a substance that inhibits respiration. So that would tell you that something was active because respiration produces the ATP. Therefore, if you treat them with a substance that inhibits respiration, then you stop ATP production, which is used for active processes. Sugars can be transported both up and down the plant. Aphids can be used to sample phloem sap, and that roots, young leaves and growing fruits will import sugars, which they will either use them for respiration or growth, or they could use them for storage. So parts of the roots, you have storage organs called tubers, like potatoes, where you store the sugars as starch. Now state all of the letters that provide evidence for the following conclusions: that it's an active process. So the answers that should leap out at you here would be the fact that when you change the temperature, the translocation goes up, and also when you add a respiratory inhibitor, then translocation stops. So the answers that you should be thinking about here should be B and C. And that sugars are translocated from source to sink. Well, the answers that you should be thinking about here should be A, F, and D. Finally, explain how mass flow of the phloem sap occurs in plants with a vascular system. Well, the idea here that you have to explain is the difference in hydrostatic pressures. So you've got a source, which we'll put in the middle of the plant, and a sink, which we might put at the bottom of the plant, although remember it can be at the top if it was a leaf or a young leaf or a flower or a fruit. All of those things can absorb sugars. So at the source you're going to have an area of net production of sugars, which will lower the water potential of the companion cell and therefore the sieve tube element, because the two are connected. Because you lower the water potential, then water is going to move in by osmosis into the more sugary solution, which has got a more negative water potential. That movement of water in from the xylem is going to cause an increase in hydrostatic pressure. Now remember, the sieve tube element is a tube, series of tubes running down with gaps in them, which are the sieve plates, and then you get to a sink. Now the idea about a sink is that it's an area of net consumption of sucrose, so that the sucrose is either being used for respiration or it's being stored as starch, and starch, remember, is insoluble, so that would raise the water potential. So either through consumption of the sucrose in respiration or storage as starch, the water potential of the cytoplasm at the sink is going to be less negative than it is at the source. And because it's less negative, then less water is going to flow in. Therefore, there'll be a lower hydrostatic pressure at the sink than at the source, because there's a less negative water potential. So you've now got differences in hydrostatic pressure between the source and the sink, so that the phloem would move by mass flow from source to sink, because you've got a higher hydrostatic pressure because you've got a lower osmotic potential at the source, whereas at the sink you've got a less negative osmotic potential because you've got less sugars, therefore you've got a lower hydrostatic pressure because less water moves in. Now all those points you could have raised in your answer. So what they were looking for was, I'll make this a bit bigger, that the sugar or sucrose or the products of photosynthesis are in the sieve tube elements, that they enter at the source, and that lowers the water potential, makes it more negative because you've got more sugars. And then the water can enter by osmosis down the water potential gradient, and that will increase the hydrostatic pressure because the volume of the cytoplasm will increase because the water is moving in, therefore it will push against the cell wall, and the cell wall, being rigid or having a tensile strength, will push back onto the cell contents, therefore generating the hydrostatic or turgor pressure. Whereas at the sink, the sucrose is leaving or being used, which would increase the water potential, make it less negative because there's less sugar. Therefore water would leave by osmosis, which would lower the hydrostatic pressure. And that therefore the sucrose would move from a high hydrostatic pressure in the source to a lower hydrostatic pressure down the pressure gradient, moving through the phloem sieve tube elements. And that concludes this paper. I hope it's helped, and good luck with your exams.